Introduction to oxidative phosphorylation and electron transport chain.
The learner outcomes towards the end of the lesson. The learners, they'll be able to define oxidative phosphorylation, understand the importance of oxidative phosphorylation. They'll be able to explain all the steps involved in the process of oxidative phosphorylation. They'll be able to narrate mainly the chemi-osmotic theory and also understand the outline principle that drives ATP synthesis.
The contents of this module includes definition of what is oxidative phosphorylation, the basic steps involved, and outlines of each of the step of electron transport chain and oxidative phosphorylation. Let us see the definition of oxidative phosphorylation. So oxidative phosphorylation or electron transport linked phosphorylation or terminal oxidation. So it is like a metabolic pathway in which all the cells, they use enzymes, they oxidize the nutrients and thereby they release chemical energy. So here the chemical energy is transported in the form of electrons, and then what you see is that finally ATP is produced. So these are the sequence of steps. What we see is that we have energy-rich compounds. These energy-rich compounds include our carbohydrates, fatty acids, and amino acids. We know that these undergo series of reactions during the process of metabolism. Mainly here we'll be talking in terms of the catabolic pathways, wherein all these biomolecules, they are converted to carbon dioxide and water. During this process of conversion or catabolism, you see that lots of reducing equivalents are produced. The key reducing equivalents of our interest, they are NADH and FADH2. So these two reducing equivalents, they enter our main thing, that is the electron transport chain. So during this process of transfer of electrons through the electron transport chain, there is some loss of energy taking place, but during this process we also see that there are protons which are pumped, because of which you see that ATP synthesis is taking place, which is our oxidative phosphorylation. And the protons, they are also coupled with this oxygen to form water. The electrons are finally transferred here and hydrogen also goes there, and finally the oxygen is converted to water. So during this entire process, the carbohydrates or fatty acids or amino acids, the macromolecules, these biomolecules, during the catabolic process, reducing equivalents are produced, and here as they enter the electron transport chain, protons are pumped, electrons are transferred, and finally you get ATP synthesis taking place. Towards the end, some hydrogen and electrons, oxygen is the electron acceptor, so it is transferred to oxygen and finally water is produced. Our interest is this one, that is how is ATP synthesized. Now the question comes, where does it occur? Oxidative phosphorylation, it occurs in the mitochondria, and there itself it is linked with the electron transport chain. Oxidative phosphorylation, it is the final step of cellular respiration, and the components of the electron transport chain, they are located in the inner mitochondrial membrane. You know that mitochondria, it consists of two membranes, an outer membrane and an inner membrane. So the components of electron transport chain, they are located in the inner mitochondrial membrane. So this is the summary of the process. All the macromolecules, carbohydrates, fatty acids and amino acids, these are oxidized to carbon dioxide and water. During this process, the NAD plus and FADH2, they are reduced to NADH and H plus and FADH2 respectively. And again, these enter the oxidative phosphorylation, giving rise to ATP. ATP synthesis is taking place. And during this process, these are regenerated. ATP synthesis is taking place by phosphorylation of ADP. So what are the main steps of this oxidative phosphorylation? So we have four major steps that we'll be learning during the next course of study. So first is the delivery of electrons by NADH and FADH2. Then electron transport and proton pumping. How is it taking place? We'll see. Then splitting of oxygen and formation of water. Parallelly, ATP synthesis. So all these four aspects we'll be studying step by step. During this entire course of study, one has to learn this chemiosmotic theory. So this is the principle by which the entire process of oxidative phosphorylation will be taking place, and it's an universally accepted one. So this chemiosmosis is nothing but it's the transfer of electrons from electron transport chain to ATP synthase, and this occurs exclusively by the movement of protons across the membrane. Okay, across the membrane, that is the movement of protons from the mitochondrial matrix to the intermembrane space. So this entire process, again, it includes two processes. Okay, one is the exergonic process, the other one is endergonic process. So these two are again coupled. One cannot occur without the other. So they are interdependent and they are coupled. So this part is the exergonic process, while this part is the endergonic process. So let me just brief you up about the process which are taking place, just outlines, okay? So the process of oxidative phosphorylation, it involves these basic fundamental concepts. First, we have to understand these things. First one is electronegativity. Next, the sources of reduced NADH and FADH2, and then anatomy of mitochondria. Now, where do we get this NADH and FADH2? What are their sources? We know that in the cytoplasm as well as in the mitochondrial matrix, you have these electron carrier coenzymes. These are free, and both of them are NAD plus and FADH2. During the process of this TCA cycle or the citric acid cycle, or even in the beta oxidation of fatty acids, glycolysis, in all these aspects, we see that these are reduced. So what you have is NADH and FADH2 are the energy rich forms of these electron carrier coenzymes, and these are transported to the mitochondria, and these provide high energy electrons to the protein complexes which are present over there in the inner mitochondrial membrane as a part of electron transport chain. We all are aware of the concept of electronegativity. So oxygen is considered to be highly electronegative. We know that. In oxidative phosphorylation, what happens is the elemental oxygen that serves as the final electron acceptor. And we all know that oxygen availability is very high as compared to any other things. So because of the increased availability of oxygen in the environment, oxygen has become the major acceptor for electrons during the oxidative phosphorylation process. The third one is ATP synthesis. So here what happens is, as this NADH and FADH2, they are donating the electrons, and as the electrons are moving here, you have this complex 1, 2, 3, 4 in the intermitochondrial membrane. As these are moving along the protein complexes, hydrogen ion deposition will be taking place in the intermembrane space. So this process only we have referred previously as chemiosmosis. So this difference in the proton concentration or hydrogen ion concentration in the intermembrane space, that is more important. It results in electrochemical gradient. So this protons, they are to be pumped only through ATP synthesis back to the matrix. So here, as they are being pumped, ATP synthesis will be taking place. So this you call it as the proton motive force, because of which ATP synthesis is taking place, because of the ATP synthases which are again present in the inner membrane of the mitochondria. So we'll be dealing this again in detail.
The reference books that are used while preparing this material are Leninger's Principles of Biochemistry, then Biochemistry by U. Satyanarayana. Also I refer to Harper's Illustrated Biochemistry.
What you have learned are the introductory concepts of oxidative phosphorylation and electron transport chain. The first thing is the chemiosmosis theory. I hope you have understood that. Where are these complexes located? What is NAD? What is FADH2? Why they are important, and how ATP synthesis is taking place. We know that ATP synthesis is taking place because of the proton gradient which is generated across the membranes. So we shall see them in the next topics. Thank you for watching this video. Please do the assessment. Complete it. Okay. Thank you.